Hi guys, welcome to lab exercise 5.2 where we are going to look at our results from last week where we did a bacterial culture swab and we're going to look at some microorganisms under the microscope. So if we look here, we are going to jump straight to retrieving our cultures. Since we are online, we are going to look at some cultures that have already been done for us. You can find these in the PowerPoint where the video is found just by clicking this bacterial culture swab. So when we're looking, this is what we should have done last week is swab the different parts of the whatever we want to test with our sterile swab and then streak it across the agar plate. So these are what some of the results might look like. This petri dish was just left open to the open air, but we are going to do a qualitative analysis. A qualitative analysis is to observe these swabs and determine how many different types of bacteria are growing, what do they look like, what's their shape, what's their color, and general information like that. So if we look here, this is the same image, but this one has been a little bit colorized, whereas this might be, the one on the right might be what it looks like naturally to your eyes. But after color treatment, you can see that there are several different distinct types of colonies. For instance, colony A here, these B colonies, and colony E here has a very different appearance. So we are going to describe the bacteria that's growing on our plate. A lobate is going to have that lobed structure. So colony A has these lobes, but the surface appears smooth. And then we also have to describe its color. To describe its color, we're going to go over here. It is a white, milky color. Colony B is round, and if you look at it, it's very pale yellow over here. This pale yellow color, and it's also smooth. It's very circular, concentric. Colony C over here, if we look on the right, it is also white, but it's a stronger white than this milky white that you can kind of see through. This one is a stronger color, and it is also circular or concentric. The last one down here has an irregular border. We could call it lobate because of the lobes, but there are other types. If we look at our worksheet, the descriptions are going to either be circular, irregular, filamentous, rhizoid, or punctiform. I don't see spindle too often. Let's look at some other examples. So this is an example of punctiform growth where it kind of is little dots all over the surface is an example of punctiform. Let's see, these are considered irregular. Here we have some more smooth bacterial growth. This bacteria kind of grew a film over the top, which is not surprising considering it was from pond water. Now, this one it has interesting results. This is the result of swabbing washed and unwashed hands. Now, you can see the distinct difference between these colonies. This colony has a dimpled texture and an irregular border. This colony has a very smooth texture, and it does have a wavy border, but is not as irregular as this one is. So you can see a real difference. But the shocking thing is that your skin naturally has a lot of bacteria on it, even when washing your hands. So the unwashed bacteria was different than the bacteria that is normally the normal flora that you find on all persons' skins. So we are going to look at a couple different ones. This one is really nice because you can see an example of a fungus growth or a fungal growth here. Colony D has this white fuzzy appearance. A white fuzzy appearance is very indicative of a fungus. And the other ones here are also going to be bacterial. You can see this irregular shape of colony A. And we're going to do some different analysis. So that was the qualitative analysis. So just based on the qualitative analysis, which one of these is going to be the most diverse? So we see very consistent growth on the chicken, and in the soil we have a little bit of a rhizoid over here on colony B. But we're just looking for the sheer number of different types of bacteria. So I would say air has the most diversity. The table also has a lot of diversity, so several different types of bacterial growth. Now, did that support your hypothesis from last week? Last week we swabbed the keys, and I had mentioned that many of the students, they swabbed different things inside the building, including the water fountain, the toilets, the floor, the keyboard, a cell phone, my sunglasses. And the results were pretty consistent. Things like the floor and the toilet did not grow a whole lot, but things that are touched very frequently, like the phone and the sunglasses, did grow a lot. So does that support your hypothesis? On this grid right here on this table, we're going to describe two different surfaces. So I want you just to choose two, either the table and the air, pond water and hands. Choose two of these, describe them, the morphology and the color. We don't necessarily have to do the size, but the size information is also available up here, right here, the first column. So describe two different plates. You only have to describe two colonies from each plate. You don't have to describe every single colony on every single plate. Just to choose two plates and choose two colonies. The next analysis that I want you to do with the same two plates is to quantify the data. How much bacterial growth is on that plate? So quality data or qualitative data is a descriptive data, and quantitative data is going to be numerical data. So what we did in class was we actually drew a grid line over the top of the plate, and we can recreate that here, I think by inserting a table, and we're just going to insert a table like this. All right, so this is my grid line, and what I'm going to do is just size it right on top of my raw chicken plate, and I'm going to count how many total squares I have. So I have 1, 2, 3, 4, 5 times 1, 2, 3, 4, 5. So my total number of squares is 25. Then I'm going to count the number that have evidence of bacterial growth, any evidence of bacterial growth. So for this, even this section counts because there's a little bit of bacterial growth underneath it. This is all 25 squares. So 25 divided by 25 is going to be 1 or 100% coverage. Lots of bacterial growth on this. But we can do this with another plate, such as this one right here, and we can put it over the top of our plate. We still have 25, but now we only have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 squares that have evidence of bacterial growth. So in this case, we will do 11 divided by 25, which is 0.44, or you could say 44% coverage. This is quantitative data. Now, notice that we're getting much different information. The quantitative data is going to tell us how vigorous the growth is after our incubation period, but the qualitative data is really important because it can help us identify the different types of bacteria that are growing. For instance, E. coli is a mutualistic bacteria that lives in the human digestive system. It helps us to absorb vitamin K, but It can also be a pathogen if it's then reintroduced or a pathogenic variety is introduced to either the urinary tract or the digestive tract or the lungs. So it can cause different diseases that way. All right, let's look at these questions. It says, was your prediction correct? Well, in our experiment in class, the most bacterial growth that was seen was actually in the water fountain, not on the handle that people touch, but the water of the water fountain. And the second most growth was going to be either the cell phone or the sunglasses, some personal item that people touch quite frequently without washing. So was that your prediction or not? How might variations in environmental conditions affect these growth distributions? Well, if it is an item that is protected from the elements, it might grow more bacteria. For instance, one student, they swabbed the concrete outside and it did not grow too much because it had rained or the environment had been too harsh. But which grew the least amount of bacteria? Well, the floor did not grow anything. The toilet seat grew some things, but not very much. So I would say those were the least active plates. The keys that we swabbed did not grow very much. I imagine because they're smooth and shiny and the bacteria couldn't grab onto them. So how might the presence of disinfectants affect these results? Well, one of the hypotheses was that the bathroom would have a ton of bacterial growth. The bathroom is obviously where you're going to find a lot of E. coli since E. coli lives in the human digestive system. But the thing about our building is that we mop the floors twice a day, every single day in the lab, and the bathrooms get washed very frequently with antimicrobials and disinfectants. So we did not see a lot of growth in areas where these disinfectants were used. So these disinfectants can be very effective at reducing bacterial growth. That being said, not all bacteria is pathogenic. So, for instance, the washed hands is evidence of bacterial growth of the normal human flora, whereas the unwashed hands may have other types of bacteria in it that could possibly be pathogenic. All right, what is the significance of maintaining sterility? Well, let's look at this experiment. If I swabbed the table and did a petri dish, and then I swabbed the chicken and did a petri dish, and then I swabbed the soil and did a petri dish, would I know what my results were from? Or could I accidentally get chicken bacteria in my soil swab? Could I accidentally get table bacteria in my chicken swab? I would never be able to know where my results came from unless I used very specific sterile techniques. For instance, this fungal growth is evidence that there might have been some contamination with that fungus, or that fungus was growing on the surface that we swabbed. So contamination of the results is our main concern with maintaining sterility. We also want to make sure that we are keeping ourselves protected from any pathogens. Now, why would we do qualitative analysis? Qualitative analysis is where we're describing the features. The value of this is that this will help us identify the bacteria. Even things like the smell and the color can help us identify what the bacteria is. This gives us really good information about the characteristics of the bacteria. Whereas quantitative analysis is a lot easier to do statistical analysis and statistical comparison. So quantitative analysis does have its place in finding out how much of something there is. Now, next, the students in face-to-face class created a wet mount slide of some pond water. What I want you to do is view this video with me about the pond water. And we can draw a picture of an organism that we see and any identifiable features of that organism. I only want you to do one or two. It's up to you how much you do. We were not going to be able to know the magnification because we're going to watch a video. So let's go ahead and watch that video. All right, you can look at many different videos online for these microorganisms under the microscope, but the purpose of our exploration is to see that microorganisms live everywhere on Earth, in every surface, and some of them can be quite large and some of them are very small. For instance, this nematode here. So what you can do is zoom through one of these videos, pause maybe. This is actually a really helpful video because it has labeled the nematodes. It labels the water bear. This is a water bear here, or tardigrade. They're very interesting little creatures. Just pick one of them and draw a small sketch of it and any identifiable features that you see. And that will go in this box. The last bit is just a reminder about how to use a microscope, which, since we are not able to use the microscope online, can be disregarded for now. The questions for this week's quiz will come directly from the information in this assignment. Thank you for joining me this week for exercise 5.2, and I look forward to seeing you next week for 5.3.